What you lost?
The clicker.
I haven't
Oh,
got it.
that is
Nancy, have you
immediate.
stolen it? Put it in your bag.
No.
Hannah was using
The
it.
man who's doing the
She
swap.
may still have it.
Clicker, technical term.
Is there a keyboard there?
No.
Probably one underneath. What else fails?
Maybe just go
Hannah
and
was
ask them in
playing
the
with it,
box.
saying it works very slowly.
Okay.
Yes, she was. But she's kept it.
Either
Give
Hannah
the
still
bigger
got it
battery.
or she gave it back to them.
You found it? Yay!
Yeah.
I think they were.
Fidgety.
On my phone. Where is it?
Because you haven't asked me to give any safety announcements. In case of earthquake, run like
You're
hell.
disappointed.
And you're excited about that.
If there's a fire, somebody will
There is no designated fire alarm at this time. So if you get a fire alarm, run. I think we're probably old enough to know how to do that.
It's hot. Do you want me to go and get people?
Yeah, it'll take 5 minutes at least to get them in.
I don't know, we're running already.
I'll start at least 5 minutes before.
Mouse nets a new level of enrichment. That's
Hmm.
kind of thing.
Oh, right.
Give them an extra
Platform.
story.
Very good.
An extra
Medicine
story.
to sort of
Yeah.
chill out of.
Promotes
Houses
natural.
and everything.
Hold on, I think we're
That's what they should say.
It should be nice. Yeah, take
But this is what they need. I mean, you could read this in a human context. Promotes natural home. Promotes natural behaviours.
do that for some of our students, please.
Provides a safe haven during potential floods.
Floods.
That's a useful thing, isn't it? That a home knows. Fully autoclavable, no home got that. Simple to move between cages.
Sounds wonderful.
Yeah.
And what's it for?
You get an extra, you put it in the mouse cage, you get an extra floor. It's an enrichment. An enrichment and a refinement after that.
The wood tweaked. They could eat it as well.
Oh, but that might affect their diet.
Oh, shucks.
Yes, wouldn't control
No,
that.
can't do that.
Yes. I like
It
it.
also says it's legal.
I'm just hard.
I'll just speed up.
So you definitely something.
I'll say it very fast. It isn't actually very fast. Don't worry. Most of it is what you're saying. One page which is about you. Which isn't enough. You deserve a book.
I can't listen to, sometimes go listen to Desert Island because they'll learn more about me than they will from me a year. It's like that.
You've been on Desert Island.
Oh,
Don't find out what you
2
chose.
years ago. Yeah, quite an eclectic choice actually. But they took about all this stuff.
Yeah.
And then just before,
See you. Sorry I missed your meeting.
Oh, that's okay.
I've only just about got off the signal. Yeah, most of it.
in a way.
It's the last week. Raring to go. It's been horrible.
Yeah, well, you're sitting at the front, so presumably. Okay. Yes. Well, I'll leave you to concentrate.
Yes, okay. Catch up with you.
Okay, good evening. Can you hear me? Yes, good. I apologise, my voice is awful. Hopefully it'll last for the rest of what I have to say, but luckily you don't have to listen to me for very much. So, ladies and gentlemen, honoured guests, I'm delighted to welcome you to the 6th Annual Openness Awards and the 83rd Stephen Paget Memorial Lecture, which will be given this evening by Professor Dame Nancy Rothwell. As the current chair of UAR's council, it's been a privilege to see just how much the Concordat has grown since it developed from 2012. 2019 is no exception. As we celebrate 5 years since the launch and reflect on its achievements, we also look to the future and how the Concordat continues to evolve and encourage even greater openness amongst its signatories. You should have got a copy of the 5th report tonight, which details all the great work our Concordat signatories have done over the last year. And every year it's a real pleasure to see good practice supporting transparency around the use of animals research and the extent to which things have changed. There are, I believe, spare copies. Should you wish to take them back and share them with your friends, we'd be delighted. And I've been reminded I should thank the MRC very much, who paid for the printing of the report. Looking back on the last 5 years, it's clear to see that the Concordat has had some big impacts within the life sciences community. Animal research facilities have greater profiles within their institutions, which has led to greater investment, more appreciation of animal care staff, and importantly, better animal welfare. There's also a greater access to information about animals in research from the organisations that carry out this work. More organisations than ever before are sharing images and videos of their research, their animal research statistics, and even details of the severity of the procedures. And for those interested in this type of work, there's much better access to see inside animal facilities, whether it's via open lab initiatives or virtual tours. And UAR has experienced fewer reactive communications on the use of animal research, I'm sure in part as a result of all this openness. Over the last 5 years, we've seen signatories collaborate with each other in the name of openness, and we're delighted the ethos of the Concordat has now spread well beyond the UK. In fact, today, the European Animal Research Association has launched a transparency agreement in Belgium based on the UK Concordat. But 5 years in, it was time to shake things up. All of our 122 signatories work hard to push openness within their organisations, but over the years we've seen some organisations driving the sector forward to be more transparent and more innovative with openness activities. We want to recognise these, so we developed Leaders in Openness, a new standard for organisations that are consistently achieving and embedding good practice. And recognising these organisations also allows us to provide clear examples of how good practice is being implemented. The Leader standard isn't appropriate for all Concordat signatories, but those who choose to apply to become Leaders in Openness must demonstrate how they meet good practice criteria, which are assessed by both the peers and the public. And after 3 rounds of assessment, which included review from the lay public, we were delighted to announce our first set of Leaders in Openness at the annual Concordat Signatories event last May. These 13 organisations will hold their status for 3 years, but nominations will be open every year for other organisations wanting to apply. You'll also notice a new section in the annual report this year. We know from previous feedback that signatories want more case studies in the annual report, so that's what we've done. You'll find detailed case studies from how our new Leaders in Openness have been meeting the criteria for this new standard. Leaders in Openness aren't to be confused with our long-standing Openness Awards. Those awards represent excellence communication in communications and innovation around the use of excellence innovation in communications around the use of animals research and the winners are organisations pushing those boundaries and leading by example. I'd like to thank all our sponsors for their support this evening, and they're listed in the booklet. I want to thank our judging committee for their time and deliberations, and they ensured that the awards we're going to give out encompass many facets of openness and meet the aims of the Concordat. And I'd like to thank you, Concordat signatories, for your continued hard work to promote openness. Without your dedication, the Concordat would not be the success it clearly is today. So without further ado, it's time to present the 2019 Openness Awards. And the first award will be presented by Professor Roger Lemon. Roger is Emeritus Sobel Professor of Neurophysiology at Queen's Square, and his main research interest is in the control of skilled hand movements by the brain, and understanding why hand and finger movements are particularly affected by damage to the cortex and its major descending pathways as a result of stroke, spinal injury, or motor neuron disease. His experiments have involved the use of purpose-bred non-human primates, since these provide the best available model for the human sensory motor system. He's carried out parallel experiments in normal human volunteers and has sought to apply the knowledge gained from his work in monkeys to the effects of stroke on hand function in patients, and to understand the process of recovery and to investigate therapies that might enhance recovery. He's actively involved in the public dialogue on the responsible use of animals in biomedical research, and particularly, therefore, the importance of using non-human primates as models of complex neurological and neurodegenerative disease. Roger's been a long-standing friend to UAR. He spent many years advising us during his time on council and our policy subgroup. And he also helped to establish the UK Primate Expert Group, and he's previously been on our Openness Awards judging panel. Please join me in welcoming Professor Roger Lemon.
Thank you very much, Jeremy, and good evening, everybody. And it's my privilege to talk about the award in the first category, which is internal or sector engagement. And I was quite fascinated by this award because it reflects, I think it does something about the problem that I think is recognised in many research institutes of how those working in animal research actually are able to talk outside of their institutions about the work that they do, and that which of course can sometimes be rather upsetting when you, for instance, go home after a day where you had to carry out various unpleasant duties, including euthanasia of animals that have already served their purpose in the research. While most people in those sorts of jobs are able to share their work ups and downs with their family and friends, I think for those working in animal research it can sometimes be quite difficult. If you open up fully to exactly what you do, that can expose you to a barrage of questioning and can actually lead to quite difficult interactions. So how do you tackle that sort of problem? How do you talk about the work that you do? Well, results from a staff survey at the winning university of this award suggested that a first step might be to actually bring all the colleagues together, all of whom are involved in the work, so that they at least could talk amongst themselves about their different experiences with animal research. So in response, the university organised a Tribute to Animals Day to recognise and respect the contribution of research animals that they had made, those animals had made, to the progress of research at that university. And generally to celebrate the benefits of that science. In a large research university, this event was a rare opportunity to bring together all those involved in the research, including technologists, researchers, all the way down to the principal establishment. It also included a section on the impact of the research in different fields, including clinical progress. Actually included a talk by an NHS research nurse, and she explained how talking to patients about the current research that was going on was an invaluable way of explaining to patients the importance of animal research. So overall, this was, I think, an enterprising idea. The judging committee thought it was an excellent example of how to show culture of care in the university, provision of institutional and wider personal support for those working there. and they were impressed to see how the whole institution, including its senior staff, had been engaged in setting up this event. And so I'm delighted to present the award for internal or sector engagement to Newcastle University. We have to pose for this photo.
I'm afraid so.
Right.
Thank you very much. Thank you.
Congratulations.
Thank you very much.
Thank you. And so to the second award. It's my pleasure to welcome Ken Appleby, who will present the second award. Having begun his career as a junior animal technician a few years ago, Ken is now Director of Biological Services at King's College London. He was a member of UAR's council for 6 years, and until May he served as Chair and CEO of the Institute of Animal Technology. He's a founding trustee of Animals in Science Education Trust and a Chair of the Board of the Trustees of the College of Laboratory Animal Science and Technology. Sorry, I had to turn a page over. And for 6 years, until May, a member of the government's Animal in Science Committee. One of Ken's ongoing major professional interests is in the profession and development of education and training for laboratory animal care staff. And in 2014, he was quite rightly awarded the OBE for services to animal technician education and animal technology. He complained that in this CV we'd left out the fact that he's a Hammers fan. I don't know whether that's relevant. Please join me in welcoming Ken Appleby.
Well, thank you very much. I've been told to try and stick to the script and no ad-libbing for reasons that people have heard me speak before. I'm presenting an award on public engagement activity, and I think some of us, as sort of hinted at earlier, who are a bit more mature, can remember how things were with the general public, say 20, 25 years ago. And I think it's a measure of how far we've come that when you read the nominations for this award, which is most welcome and in fact makes things in a working environment, and not only for us, but it gives the general public an in on what we actually do. And I think every time we show people around our facilities, I think they're quite amazed at the efforts we go to, both for animal welfare and for good science. The judging committee was impressed with this university's approach to a public engagement event. This event was held in a public space and was truly for the public, as it was open to anyone who signed up. They created a dedicated time and space where those who were interested could ask questions about animal research and welfare. The seniority and profile of the staff and the researchers who took the time to get involved with this activity and talk to the public about their work was a striking feature of this event. The judges felt that a good mix of science and welfare was presented, but what made this activity stand out was a dedicated question and answer section, something which saw the staff involved deal with some complex and interesting questions from the public. None of this was screened or without any prior knowledge of what questions would be asked. Therefore, I'm thrilled to present the award for public engagement activity to the University of Edinburgh.
Third award of the evening is going to be presented by Professor Rothwell, tonight's lecturer. I'll introduce Nancy more fully later before her lecture, but for now, just join me in welcoming her to the stage.
Thank you. It's a great pleasure and a privilege to be making this award for media engagement or media stories, something that has become more common but at one time was almost unheard of for anybody to go onto television or radio to talk about animal research. And it's still quite a brave thing to do. So this year the judging committee was impressed by one particular media story. The winning organisation proactively invited a BBC journalist to its animal facilities to record a radio broadcast. The judges felt that the researchers involved in that broadcast spoke informatively and sensitively about difficult topics such as cervical dislocation, giving mice lung cancer, and the numbers of animals used. Not easy topics to tackle. The judges were particularly impressed by how the researchers answered these difficult questions, as well as speaking realistically about the benefits of the research and the differences and similarities between mice and humans. This is indeed a truly excellent piece of work. So please join me in congratulating the winner of the Media Engagement or Media Stories Award, the Francis Crick Institute.
Our next award will be presented by Ross Millard. Ross is Managing Director of Agenda Resource Management Limited, having joined them in September. During his 18 years within life sciences, he's worked with numerous universities, government-funded bodies and charities to facilitate improvements in staff working conditions, animal welfare provision and consistency of research. He's Treasurer of the IAT Oxford branch, a position he held for nearly 10 years. Please join me in welcoming Ross.
Thank you. It's a real pleasure to be presenting the award in the category Website or Use of New Media. Talking about animal research statistics and putting these big numbers into context can be difficult. But the judging committee were impressed that this university found an innovative hook to promote its statistics. Thanks to well-made infographics, this media campaign was clear, engaging, and the data was not sanitized. This was a brave piece of work that reached a lot of people. It's not every day that a Concordat signatory can coordinate an animal statistics campaign around the arrival of a cute baby llama. But this university seized the opportunity perfectly. Please join me in congratulating the winner of the Website or New Media Award, the University of Reading.
Final award this evening is going to be presented by Wendy Jarrett, our Chief Executive. Wendy has 30 years of experience of communicating complex and sometimes controversial issues to the public. Whether that's important for this particular speech, I'm not sure. She joined one of UAR's predecessors, the CMP, in 2004 and worked on merging with the Research Defence Society to form UAR in 2009. Following a brief spell at NICE, she returned to UAR as Chief Executive in November 2012. She's Vice President of the IAT and she sits on the Animals in Science Committee. Please join me in welcoming Wendy.
Thank you, Jeremy. I want to say again that we are really grateful to our judging panel for the annual Openness Awards that you've just seen. We know they sometimes struggle to decide on the winners because it's we do get a really high quality of entries, and it is a bit of an invidious task, so I'd like to take the opportunity to thank them again for their time and for taking it off UAR's shoulders and onto somebody else's, so thank you very much for that. But each year the UAR team presents its own award to an individual who we feel has consistently gone above and beyond their work to support openness in animal research. This year, the more astute among you will have realised that we couldn't decide on one person, so we're actually giving 2 awards. Alphabetically first is somebody that many of you will know, but it's probably not a household name. The name of the organisation they work for has changed a few times over recent years, but they have consistently worked to help the public understand why animals are used in research, and in particular how research animals are bred and acclimatised for life in a research facility. This person has organised media visits into their facilities, both reactively, following really quite nasty negative media coverage, and then proactively, in order to help prevent future misleading media stories. They've also worked behind the scenes to try to improve openness right across the sector. I'm really delighted to present the first of this year's individual Openness Awards to Val Summers. The other person we'd like to recognise this year has also been prepared to work with the media in both reactive and proactive situations. They've helped us as an organisation on numerous occasions when we have been looking for somebody who can talk to journalists, to MPs and others about the realities of animal research. They've helped us to develop the Concordat, and they continue to support it wherever they can. They also work on behalf of the sector as a whole, liaising with the Home Office, chairing meetings, giving media briefings. I'm amazed sometimes that they manage to do all this, as well as a high-level day job aimed at treating a horribly cruel inherited condition. Our second individual award this year goes to Professor Nick Wells.
Thank you, that's so unexpected. Thank you very much.
No. Just, I don't know where it is. Oh, there it is. She's got it. So, to the Stephen Paget Memorial Lecture. This lecture celebrates the life of Stephen Paget, who passionately believed that a greater understanding of physiology would lead to better medical advances. He was the founder of the Research Defence Society, which later became UAR. And this year we're delighted to celebrate the 83rd Paget Lecture, adding Professor Dame Nancy Rothwell to our long and eminent list of lecturers. Nancy is President and Vice-Chancellor of the University of Manchester. Alongside her outstanding past and present professional achievements, which you can find in today's programme, Nancy was also chair of the Research Defence Society, UAR's predecessor, from 2004 to 2007. She just told me she didn't know when it was, now you know, okay? Nancy maintains an active research group. For the last 3 decades, her research has concentrated on neuroscience, spanning from cell biology and animal research right through to clinical work. Animals have always played an important role in her research. Her early work focused on weight loss and metabolism in disease, but when she discovered that blocking cytokine action could reduce not only metabolism and weight loss, but also brain damage in an animal model of stroke, she switched field to concentrate on stroke research. Her current research focuses on the role of inflammation in brain disease and she's identified the role of the cytokine interleukin-1 in diverse forms of brain injury. Her recent studies have begun to elucidate the mechanisms regulating interleukin-1 release and its action, and her group is currently conducting clinical trials of an interleukin-1 inhibitor in stroke. Since somebody in the UK has a stroke once every 5 minutes, we hope that this will be successful and lead to benefit to patients from her work. So I'm delighted to introduce Dame Nancy Rothwell for her lecture, A Stroke of Bad Luck. Nancy.
So thank you very much indeed, Jeremy. I don't need to give my talk now, you've summarised it perfectly. It's really a great honour to be invited to give the Stephen Paget Lecture. I hosted the lecture, of course, for 3 years when I was President of the Research Defence Society. Now I know the dates. Thank you. I'm going to give you a very brief background of my research on animals in particular, with the main focus on our research on stroke, particularly addressing the issue of the relevance of animal studies in that and translations to patients. And I'll briefly mention the importance of public engagement, which has been a passion for me since I was a PhD student. Apparently, when I was 2, I declared that my goal in life was to be a famous scientist. This is me age 2, avidly reading It's Lancashire Life, actually, not a scientific journal, but then I forgot about that. You've heard a little bit about the background that's relevant to this lecture. I conducted animal research as an undergraduate in my project, a PhD student and beyond, and I've done many presentations on the value of animals in research, including public lectures and television. I was the director of the University of Manchester Animal Facility for many, many years, and I'm delighted we were one of the first to sign up to Concordat and indeed invite many visitors into it. And then you've heard some of the other things. I was the founder of the Biosciences Federation Animal Welfare Group, and that then became the Royal Society of Biology, a very active group. And I was chair of the National Centre for 3Rs Grants Committee. And I do want to acknowledge many organisations who contributed to the position we're in today of a very different approach to animal research and to animal welfare, including, of course, UAR, those that have been mentioned, and the National Centre, which has done a great deal. So you heard briefly that I got into stroke research entirely by accident. My early research with Mike Stock, and by the way, my teeth were different because he broke them playing squash one day, as your PhD supervisor does. And we were interested in how body weight is regulated. Why do some people get obese and some people stay lean? And indeed, we were looking at it slightly differently because, of course, everybody is aware of energy balance in mammals, in animals, as well as in physical settings, where energy intake must either equal energy expenditure, mostly as heat, or there will be a change in body energy content. And the predominant view at the time was that obesity was largely as a result of excessive energy intake, and indeed above expenditure, which of course it must be. But what we were interested in was whether also energy expenditure could be regulated, such that higher levels of heat production could allow some animals, and indeed people, to maintain weight even when intake goes up. And indeed, I was the subject of an experiment during my PhD when we had to eat 5,000 calories a day for 3 weeks. I did gain weight. I gained about 5 kilos and lost it again in the following 3 weeks. There are members of that study who were obese from the moment it finished and are still obese today. And actually it's got worse. And so they don't thank Mike Stock for that. But largely what we did was to study thermogenesis, and in animals it was recognised that a specialised tissue called brown fat can help them keep warm in the cold through high metabolism. And we wondered whether humans could do the same. And indeed, this was then at the time the subject of a BBC Horizon programme called The Fat in the Fire. And before the programme went out, and this was 40 years ago, there was a big debate as to whether we should show all of the scenes or not. And indeed, we did, and we did, of course, get complaints and lots of hate mail about it. Not about me shopping, but of course about the use of animals. This is Deirdre, by the way, who ended up being a pet rat. But nevertheless, we continued with that research, and then a few years later, I decided I was going to move from Manchester to Manchester, where you may have gathered I'm from the north of England, and then went on to a chair in physiology. and decided that I had to carve out something of my own research career. That rather than just working on obesity, I had to work on something slightly different. So as a metabolic physiologist, which is what I knew all about metabolism, I didn't change completely. I decided instead of working on why do people get fat, I worked on cachexia, which is why is it that in disease there is such extreme weight loss. And to cut a long story short, we did studies on animals and on patients that demonstrated, as had been shown by others, that there is a very high metabolic rate in many diseases, particularly when you think that patients are often eating less than they would do normally. And of course we know this from the key hallmark of disease, and particularly infection, which is fever. And that fever is achieved, the rise in body weight, partly through reducing heat loss, but also through increasing energy expenditure. And this is where the chance experiment came along. We had an animal model of stroke that a colleague was using, and I had a hypothesis that the high metabolism in these animals was due to inflammation in the brain. And so we used a blocker of inflammation. And indeed, the metabolism was reduced and the animals lost less weight. And I wrote this up later in a series in Nature called Changing, and it was called the one last control experiment, because the last control experiment we had to do was to check that the intervention was simply blocking the high metabolism and wasn't reducing the damage caused by the stroke. And of course we found out that it dramatically reduced the damage caused by the stroke. So within a period of 6 weeks, I decided to change fields completely, to move away from metabolism and into neuroscience and stroke. And from being a platform invited speaker at conferences, I ended up having to pay and go and learn and reread my undergraduate neuroscience textbooks. But for anybody considering changing fields, it's hugely invigorating to change because you have no baggage with you whatsoever. So I started working on stroke, and as you've just heard, this is a massive clinical condition. Across the world, it's the second leading cause of death, and it is the leading cause of neurological disability. And we have got a little better at treating stroke, but we're still not particularly good. So down here, this is stroke, here is deaths, and here are all the other neurological conditions. So very briefly, and I recognise you're a mixed audience of eminent and very knowledgeable scientists and those with less background, so I'll dip in and out of things. In case you're not familiar, stroke is simply defined as a reduced blood flow to part or parts of the brain, and critically therefore reduced oxygen. In the UK it is actually the third greatest killer. And it can be caused by a number of different factors. There can be blockage of an artery supplying a part of the brain, that's the most common, as depicted here. And the territory supplied by that artery then has reduced oxygen. It can result from a brain hemorrhage, an artery bleeding. It can result from poor general circulation, from heart failure, from respiratory failure, and of course in babies from birth asphyxia quite commonly. And this is a condition where during birth normally there's restricted oxygen supply to the fetus, often resulting in severe impairments. So what are the current treatments we have? Well, actually very few. The key one is this one, thrombolysis, using an enzyme, a tissue plasminogen activator, to literally disperse the clot. And the result, if you can see, here's an occluded artery in the brain of a patient, and here after TPA, a reperfusion. When TPA works, it is unbelievable. It is amazing. And indeed, when it was first licensed, there was a consideration of how the story of TPA would be promoted in the USA, because many patients who came into hospital with a stroke, they didn't rush in. And so you may have heard that it was the story of a patient given TPA was shown on the medical drama, TV drama, ER. And there was a patient who came in with paralysis down one side, couldn't speak, gave them TPA and walked out an hour later. But the trouble is that episode of ER was shown in the UK before TPA was licensed. So lots of people who slept on their right arm or something were coming into hospital. But nevertheless, TPA has transformed the acute treatment of stroke. More recently, mechanical movement of the clot, endovascular thrombectomy, is also showing very positive signs. But we have to bear in mind these treatments are available to less than 20% of patients. And there are two reasons for that. If there's any uncertainty about whether it may be been a bleed rather than a clot, then you don't obviously give something that's going to unblock the clot. And secondly, because there is a time frame during which TPA is effective, and it's really only up to about 6, possibly 8 hours. And many stroke patients don't even know when they had the stroke. They may have woken up in the morning, or they may have been on their own. So we're still left with a problem, with stroke as a massive burden. What we have had advances in is stroke, specialised stroke units, managing stroke, reducing infections, giving treatments that limit the cause of that stroke, such as aspirin, treatment for high blood pressure, and so on. But honestly, where we are, it's not in a particularly good place. It is, as I mentioned, a massive clinical, social and economic burden with limited treatments, major failure of clinical trials, as I'll come on to in a moment, little interest now from major pharmaceutical industry, and very, very low government funding. If you look at the funding per person who's had a stroke relative to some other diseases, it is one of the lowest. So why? Were they the wrong targets that were being attempted, the wrong animal studies, the wrong patients, the wrong trials? And the answer to that is probably yes to all of them. There has in recent years been a really systematic approach to animal research on stroke, and I do want to acknowledge the work of Malcolm MacLeod from Edinburgh, who's done a great deal of work on preclinical studies. And he published this quite some time ago now, tests in vitro and in vivo of potential treatments, over 1,000, tested in animals, over 600, the numbers are even bigger than this now. Effective in vivo, nearly 400, that's nearly 400 different treatments were effective in animal studies. One came through positively in a trial, and that's TPA. So that does start to beg the question, which I've thought about many, many times, are we doing the wrong animal studies? And I think I can say in this audience that it's something we should question all the time. Are we doing the right animal studies? Have we got the right animal models? So what went wrong? I think there are a number of things that went wrong. There was a focus on the brain. Why not? Stroke is a neurological disease, but actually it's a cardiovascular disease. Almost always it originates outside the brain. The fact the organs affected is the brain is secondary. Secondly, and I'll come back to this, there was a tendency to focus on disruption of normal processes rather than on specific disease processes. What I mean by that is much of the research was looking at the massive disruption to the release of neurotransmitters, like glutamate, to changes in ion concentration inside cells, like calcium and sodium. These are all critical for normal brain function, but they're incredibly well controlled. And in a stroke, there is a massive disruption, thousands of fold change. When you step back and think about it, the task of blocking that thousands of fold change while leaving that little bit that's critical for normal function intact is a huge undertaking. And in fact, all of those approaches failed. There is no doubt that the animal studies had been lacking. Failure to account for comorbidities, failure to properly mimic. Nearly all studies on stroke were done in young male Sprague-Dawley rats or mice. So that in itself is a limitation. There is undoubtedly limitations on clinical design because stroke, up until recently, was treated like one disease in the way we used to treat cancer. Now it's categorised much more and trials are much more sophisticated. But coming back to the normal versus the disease processes, we thought about, well, what is a disease process that is unimportant in normal function? And of course, the classical one is inflammation. This is from the front of Time magazine, the secret killer, because of course inflammation is well recognised to contribute to arthritis, inflammatory bowel disease, and so on. But we now realise that inflammation is a key feature, if not a key cause, of a hugely diverse range of diseases, including, interestingly, obesity. And that's changed since the time I worked on obesity, because we now know that fat cells are not dormant storages of lipid. They're very active sites of inflammation. And that is probably one of the factors that contributes to the high rates of stroke and heart disease in obesity. It's not just carrying that weight around, or not even just filling up arteries. It is because they're a site of major inflammation. And of course, atherosclerosis, key cause of stroke and cardiovascular disease, is also very much associated with inflammation. Now, we've known about inflammation for thousands of years. We've known about the pain, the loss of function, the redness, the swelling, and the heat. But it's only recently that we've started to recognise that this is important in just about every disease that we are aware of. Now, of course, inflammation is a host defence response. It's something really important It diverts blood to tissues that have either an infection or an injury. It diverts white cells. It activates the immune system. But when inflammation is inappropriate, i.e. it's too severe, it's prolonged, or it's in the wrong place, it's massively damaging. And indeed, it is the cause of many of those diseases. And we know that many of the risk factors for stroke are inflammatory, whether it's chronic inflammatory disease, recent infection, or surgery, and this builds up an inflammatory response. Key to inflammation is a whole range of inflammatory molecules, and this is where I had to go back and learn undergraduate immunology again, which is a task in itself. And of course, there are many, many inflammatory molecules. There's complement, chemokines, there are prostanoids, and so on. But particularly important are the cytokines, and these are interesting, the cytokines, because they're normally produced by immune cells, not always, and they are released in the brain during injury. But a key feature of most cytokines is they have little or no role in normal physiology. There have been some suggestions of minor roles, but really not particularly significant. The first cytokine that was ever discovered was known as the endogenous pyrogen. Remember my reference to fever, because it was recognised that since a bacterial infection didn't have to get into the brain to cause that increase in set point that caused the fever, there must be some signal from the infected site to the brain. And so there was a long search for that endogenous pyrogen. In the end, actually, it was isolated from many litres of urine of nuns in a convent who all got an infection at the same time. And they isolated the cytokine that was then known as interleukin-1. And it is still considered the first cytokine, and by some, particularly those who work on it, the master cytokine. But actually, even interleukin-1 is itself a huge family now of many molecules. And I'm just going to simplify it by presenting the major ones. There are two agonists that activate cells, interleukin-1 alpha and interleukin-1 beta. There are others, but these are the main ones. And rather fortunately for us, there is a naturally occurring interleukin-1 receptor antagonist. As far as I'm aware, it is the only example of a molecule that does absolutely nothing, as far as we can tell, apart from antagonise the actions of another endogenous molecule. Interestingly, these are all formed as precursors, which for interleukin-1 beta, it's inactive. The others are active. But this is very important because it has to be cleaved by an enzyme, caspase-1. Caspase-1 is involved in cell death. And when the C. elegans genes for cell death were identified, one of the first things that people were doing when they found the C. elegans death genes was to say, are there any similar mammalian genes? And they found caspase-1, and it does have a role in cell death. And they act on a receptor. There are a number of different receptors. There must be more than one functional receptor, but it's not known the nature of those as yet, with a very complex intracellular cascade. And there are loads of regulations on interleukin-1, things that regulate the synthesis, regulate the cleavage, block the receptor, release the receptor, block the downstream, which you would expect of a molecule that is potentially damaging. So when you activate IL-1, key is that it's deactivated. It tends to be produced very, very locally indeed. In fact, you almost never find interleukin-1 in circulation. It's just in the tissue that is damaged, unlike other cytokines. So I'll give you a few examples of this, but to cut a long story short, after that first serendipitous experiment, when the brain damage was reduced by giving, actually, this interleukin-1 receptor antagonist after stroke, we did a whole series of studies. And what we found was that there's rapid production of interleukin-1 in the brain of animals after a stroke, that administering interleukin-1 increases the damage caused by a stroke, and that blocking IL-1 markedly inhibits injury. And I'll just give you a few examples of those in some data to illustrate. So in rats, we've used a number of different species. In rats, we looked at the expression of interleukin-1 and found it occurred very early indeed. And it's shown here and interestingly, it was mainly IL-1 alpha, unexpectedly, the red staining here. And it actually all co-localizes with microglia, one of the forms of glia in the brain. And it is actually on the contralateral side as well as the side of the stroke. We administered interleukin-1 to animals to determine if that had any impact on the stroke. And this is assessed either histologically or through MR imaging to determine the size of the stroke in the brain, in just the same way as you might use MR imaging in patients. And on the left is animals given vehicle, on the right given a very low dose of interleukin-1. Giving an exogenous agent and seeing an effect in itself proves nothing, of course, but this could be very important because one of the things that makes a stroke a lot worse is an infection. In fact, pretty much all neurological disorders are worse during infection. Now, we can't say for sure, but that might be because there is more interleukin-1 being produced. Patients with multiple sclerosis in particular have much worse symptoms, and patients with dementia also do. But obviously the more important experiment, which I've mentioned before, was blocking endogenous interleukin-1, and for this we used the endogenous interleukin-1 receptor antagonist. And here is the total damage in vehicle treated, and here markedly reduced, and it reduced it in both the cortex and the striatum. Now, of course, it is possible that the antagonist is doing something else other than just blocking interleukin-1. So we used a whole range of other approaches with antibodies, with inhibitors of the enzyme caspase-1, probably most definitively using knockout animals, and these are now mice, in which the genes for interleukin-1 alpha or interleukin-1 beta or both were deleted. And what you see here is if you delete either IL-1 alpha or IL-1 beta, there is no effect on the damage because they compensate for each other. If they're both deleted, there's a marked reduction in damage. So this and many other studies by us and other people did suggest that endogenous interleukin-1 in rodents at least mediates a significant part of the damage caused by a stroke. An experiment we did which led to a result we hadn't expected was to see what happens when you delete the gene for the naturally occurring interleukin-1 antagonist. And what we found there was the damage is much worse. So in other words, that naturally occurring antagonist is serving to limit the effects of IL-1. It is actually putting a brake on it. And when you look for mutations in the IL-1 family genes and the relationship to stroke or worse outcome, actually most of the mutations you find are in this antagonist. And it's likely that the antagonist, which is produced after the interleukin-1, serves to limit its effect, both spatially and temporally. So then, of course, the big question is, how does it work and where does it work? One of the problems in treating stroke is getting drugs into the brain. Although there is a disrupted blood-brain barrier, it may not be enough. So is interleukin-1 just acting very locally, so we'd have to get the antagonist in very locally? Because although it is effective when given peripherally, large doses are needed. So we did some studies to inject very low doses of interleukin-1 peripherally, systemically, in mice, and found that even at doses, this dose here of 10 units doesn't even cause fever, but it does worsen brain damage. And this is actually neurological deficit, which is also worse. So this suggests that even very small doses, you cannot detect that interleukin-1 in circulation, can have an effect on the brain. How is it doing that? Well, we think that one of the ways it's doing it is by mobilizing neutrophils from the bone marrow. And the reason we think that is if we do this experiment and then give an antibody to neutrophils, the effect is lost. So here's the brain with lots of neutrophils in it, in animals treated with interleukin-1, here with the antibody none. Here's the exacerbation by interleukin-1, and when neutrophils are blocked, then the damage is not exacerbated. So it looks as though tiny doses can exacerbate the stroke by mobilizing neutrophils. But that suggests it can work peripherally, not it does. So the big question is, what about endogenous interleukin-1? Is that mainly peripheral, outside the brain, or is it mainly in the brain? And in some heroic experiments by Dame Nancy, when he was a postdoc, he did adoptive transfer, bone marrow transplantation. So what he did was he took classical way of doing this, mice that have interleukin-1 and mice that have none, irradiated them, and then did a bone marrow transplant. So you end up with mice that have got IL-1 in the brain and outside the brain, ones that have got it only in the brain, ones that have only got it outside the brain, and ones that haven't got any. And the surprising result was that actually in all of them, the damage was reduced. So it suggests that both peripheral and brain interleukin-1 contributes to that damage. And actually, the reason for this might be because a major target for interleukin-1 is endothelial cells, the lining of the blood vessels. And this, and I apologize, it's a slightly complicated slide, the work done by Emmanuel Pinto, he did targeted deletion of the receptor only on endothelial cells in animals. And what he found was deleting it here just on the endothelial cells caused a reduction in damage. This is just immune staining as well, and this is neurological score. So in other words, a key target for interleukin-1 is actually the blood vessels. We know there are other targets as well. So from our studies, it was shown by us and many more that inhibiting IL-1 through a number of different means inhibits damage caused by focal, very localized, or global cerebral ischemia or stroke, whether it was permanent or reversible, experimental traumatic brain injury, chemical damage caused by cytotoxins, animal models of multiple sclerosis, seizures, actually not on here, but animal models of birth asphyxia, and in animal models of dementia. In the triple transgenic Alzheimer model, it reduces. The mechanisms, which I'm only going to say very, very briefly, appear complex and multiple. So what we think is that in the brain, at least, most interleukin-1 is produced by microglia. It does act on astrocytes to release toxins from astrocytes locally. Interestingly, the effects on neurons are probably beneficial, but it also acts on the endothelium within the brain to cause activation, production of adhesion molecules, and release of chemokines. But also, there is a peripheral inflammatory response to a stroke, which you can see an acute phase response to a stroke, and that, in turn, activates bone marrow to release neutrophils, which IL-1 mediates, and again, appears to act to worsen damage. And in terms of downstream mechanisms, I'll say no more than this. That's enough, probably. But that's all well and good. But you know, do these studies have any meaning for stroke patients, particularly given what I've said about everything else has failed? And again, Malcolm McLeod did some interesting studies where, if you can't read it, he did a meta-analysis of all the experiments done with interleukin-1 receptor antagonists. And he said the animal data supporting IL-1 receptor antagonists as a candidate drug for stroke are limited, and further experiments are required before a trial is undertaken. Unfortunately, we'd already undertaken a trial, but nevertheless, we went back. Efficacy at later time points in animals with comorbidities, and it does say it's more effective when given into the brain than peripherally. So that prompted a whole series of studies, if you like, going back and saying, we need to stop just using young male rats and mice, and we need to much more closely mimic the clinical condition that happens in a stroke. And I'll just illustrate a few of the things that we did. So this is actually showing that when given quite delayed, it's still effective. So this is when it was given 6 or 8 hours later, and it's still effective. And actually, you probably can't see it on here, but these are lean animals, and these are corpulent, which I'll show you those in a minute. So it was effective when given at a time after the stroke that could be clinically relevant. We also then found that it provides long-term recovery, because a concern is interleukin-1 might be causing the short-term damage, but maybe it's good for repair and recovery, and you don't want to block it. But actually, what we found, and this is giving interleukin-1 either at the time of the stroke or 12 hours later, then looking 28 days later. This is the key graph here. So this is looking at success in the neurological score. Controls here, animals given IL-1 receptor antagonist are still improved and almost back to normal nearly 30 days later. And actually, it seems that not only does IL-1 receptor antagonist limit the damage, and that's long-term, it actually may promote repair and recovery, because it appears to drive neurogenesis, the formation of new neurons, which does occur, although not in large amounts, in the brain after an injury. And obviously, the younger the animal or the person, the more likely it is that they're going to have neurogenesis. So this shows a placebo, an IL-1 receptor antagonist, and signs here of new cells being formed, and when they're counted, the IL-1 receptor antagonist was causing a significant increase in neurogenesis. So this is, of course, reassuring, but still doesn't answer a number of significant questions that you might want to answer if you were going to take it into clinical trials. And in terms of clinical relevance, we looked at comorbidities beyond the obesity, timing of administration, which I briefly told you, pharmacokinetics, is it going to get to the place which is going to be effective in the right concentrations? Is it effective with existing treatments? Does it work when given with TPA? And how do we improve the design of clinical trials? Because otherwise, this might be yet another failure in the long list in stroke. So in terms of comorbidities, when you think of many people who get a stroke, they are often obese, they may have diabetes, they often have atherosclerosis, hypertension, infection, and they are largely old, not all. And indeed, we are actually looking at intracerebral hemorrhage and subarachnoid hemorrhage, which tends to occur in younger people. So I won't show you all of the data, but we've modelled all of these in animals and looked at obese animals, diabetic. We've also looked at animals with severe infection. And in each of these cases, the interleukin-1 receptor antagonist is effective. And just to give you one example, here's the corpulent rat here, very corpulent, and the lean one here, which caused a bit of problem looking after them. This is the damage in the lean, and this is in the corpulent, and the lower one is animals given the R1 receptor antagonist, and it was just as effective. And actually, we then had a big argument about what dose you would be giving to a corpulent rat, which is 2.5 times the size of its lean litamate. So we actually dropped it to the same absolute dose, and it still was protective, rather than adjusting for the greater body weight. But then, and I think importantly, what we did was a large cross-lab study, funded by the European Union, I have to say. I'll go no further on that. It was we had a long series of European funded research between a number of stroke labs, and we decided what we had to do was each do the experiment in our setting, in our animals, completely blinded. So I should have said that now, following all the guidelines, every experiment is double-blinded, it's fully randomised, everything is you need to do or should do. And having got involved in clinical trials, there's a lot that you could take back to animal studies that is routine in clinical trials. So what we did was a study in Budapest, 2 separate studies in Caen, 1 in Lubeck and 3 in Manchester, different investigator for each study. There were different strains of rats, but you'll see the meta-analysis showed a significant benefit for treatment. So I think that does suggest that it is robust, and I think now we have done multiple laboratories. We've done male and female animals, we've done old animals, and we've done comorbidity. So this does suggest at least that the data are robust, and thankfully Malcolm McLeod was happy. Did a systematic review and meta-analysis of the efficacy, and he says the only major standout remaining evidence required is efficacy in hypertensive animals, which has been done on the basis of R1 of evidence currently available. R1 receptor antagonist is an attractive candidate for clinical trial. Yay! So, how does this relate to clinical studies, and which we've been doing for quite some time? One of the questions that's been around for a long time is inflammation a cause of stroke? Never mind what happens afterwards. And actually, there's good evidence that is the case. But we also looked at biomarkers for inflammation and risk factors, pharmacokinetics, trial design, and patient selection. So there's good evidence that there are inflammatory episodes throughout life, whether it's a bout of flu or respiratory infection, and that each contributes to a growing inflammatory load. We all get more inflammation as we get older, whether it's a slightly creaky joints or slightly more don't recover quite so well. But there was good evidence, not from us, some very good epidemiological studies that showed in older patients who suffered from an upper respiratory tract infection, they had a 3-fold greater risk of getting a stroke or a heart attack in the 3 weeks after the infection. Now, there could be all sorts of reasons for that, but an inflammatory dive might well be one of them. And in fact, a lot of research has been done looking at inflammation in the brains of patients, either with stroke or with other conditions, or at risk. So this has involved post-mortem analysis, looking at the expression of cytokines and other inflammatory factors in their brains. It's involved microdialysis or sampling of brain tissue, but obviously some of the most powerful approaches are to use imaging. And one example that I'll give you here is using PET and MR imaging. So it's a stroke patient. The stroke is here. this is using a well-known tracer that binds to activated microglia. There are newer ones now that are being used. This is PK1195, and microglia are the main source of interleukin-1, and you see the concentration here of activated microglia around the site of the stroke. It's circumstantial, but it does suggest at least, and there have been a number of other studies done, and indeed in dementia as well, but it does at least suggest that there is inflammation in the brains of people who've had a stroke. We did some very small studies looking at patients at risk, and we defined risk as having significantly raised levels of a key inflammatory marker, but without any neurological symptoms and any obvious ongoing infection. And the marker we used was C-reactive protein. It's a good long-term marker of inflammation. And this just illustrates, it's not definitive by any means, again, PET imaging in 4 patients with high CRP and 4 with low to normal. These were all older patients in their 70s. Now, what we need to do really, but it's unbelievably expensive, is to get a larger cohort, then ask, do the ones with the high inflammation go on to get either dementia or stroke? But the cost of doing that in PET in all of them, and then a long-term follow-up, is absolutely huge. But in terms of taking what we had done in the lab in animals into patients, we felt we had 3 key questions to answer. These were the pharmacokinetics. How much do we have to give? Does it get into the brain? Does it need to get into the brain? Is it safe? And is it effective? And I'm going to give you the answer to the first 2 and only the half answer to the third. I'll warn you now, we haven't got quite to the end of the story. So pharmacokinetics. We first did some studies where we PET labelled interleukin-1 receptor antagonist, and animal studies suggested that it did get into the brain when you do the imaging. But we wanted to do it in patients, and that's quite difficult to do. So we decided that rather than look at stroke patients, we'd look at patients with subarachnoid hemorrhage. Now, this is obviously bleeding in the brain. Significant proportion of those patients go on to get a secondary stroke. About 20% get delayed cerebral ischemia in the 2 or 3 weeks afterwards. And this just illustrates an aneurysm here, which often bursts or starts to leak, causing a hemorrhage with damage here or here, and it's actually not that different. One of the differences, though, between patients who've suffered subarachnoid hemorrhage and those who've had stroke is, firstly, they're very intensively managed, normally on a neurosurgical intensive care unit, whereas stroke patients until recently have been on general wards, but also that many of them have an extraventricular drain put in. That means the CSF is drawn off because the pressure in the brain goes up because of the swelling, and therefore that has to be relieved. So a drain is inserted into many of these patients, sorry about the gory bits, drilled in through here, and this is withdrawn. So we thought this routine sampling could be a way of testing whether or not the interleukin-1 receptor antagonist got into the brain. Now, we had to do this after the initial bleed, and we had to check that there wasn't still a bleed ongoing, because that would just be coming straight in, obviously. What we found in these patients was that if we looked at plasma, levels go very high very quickly. This is the levels in the CSF. Now, the concentration required in animals to be effective is about here. So enough was getting into the brain. So that suggests that we could get enough into the brain. In these patients and in others, we have surprisingly seen almost no side effects of interleukin-1 receptor antagonists given for at least for 2 to 3 weeks. The one we were worried about was opportunist infections, particularly in these very sick patients on neurosurgical intensive care, but really saw either minimal or no increase in infection. So we've done a number of clinical studies now in stroke and subarachnoid hemorrhage, and I'll just highlight a few of those. In taking it into patients, we knew it was very safe because this drug is actually used to treat rheumatoid arthritis. It's not particularly good compared to, say, TNF antibodies because it's got quite a short half-life. But actually for stroke, you want a short half-life. For chronic treatment, you want a longer half-life. But there were really very few side effects reported, and there are trials now ongoing with inhibitors of IL-1 in myocardial infarction, in birth asphyxia, in a number of other conditions. We'd done preliminary clinical studies, so we had an idea of the feasibility of giving it, and it was given intravenously in the first study. Later it's been given subcutaneously, and we've done pharmacokinetics on that as well. So the first study we did was a phase 2 study, really just to determine can it be given and is it safe. And can we get any indication from biomarkers that it was actually blocking inflammation? It was a fairly small study. This was a number of years ago. So we chose to study a range of biomarkers in the stroke patients. And these are all markers of inflammation. And all suggested that the IL-1 receptor antagonist was indeed reducing the levels of inflammation, which was positive. So if we just look at these 4 graphs, this one gives neutrophil count in blood, this is total white blood cell count, this is C-reactive protein, and this is interleukin-6. Interleukin-6 is interesting. It's another inflammatory cytokine. You find lots of it in circulation. It's downstream of interleukin-1. And one of the reasons we know that interleukin-1 doesn't worsen damage just by increasing temperature is because interleukin-6 also increases temperature, but it's actually protective. It's the opposite. But in each case, the first thing you see is this acute phase response to the stroke. The open triangles here are the stroke patients given placebo, and in every case, there's an acute phase response, which is probably worsening the damage. And in each case, the patients given the IL-1 receptor antagonist, the active component, it was largely reversed. It was far too small a study to be powered to get any definitive outcome, but we did look at neurological scores, and these are 2 different scores. And in each case, the white part is fully recovered, the black is dyed, this is lost. And on both measures, it did appear that there was more patients in the IL-1RA treated group that did well. We've then done a larger study with biomarker outcome, and we used interleukin-6 and CRP, the area under the curve, in patients treated with placebo or the interleukin-1 receptor antagonist, and in both cases, it met the primary endpoint. We then did a larger study of in acute stroke, and it met the primary endpoint of reduced markers, but there was no clinical benefit. So we asked, what's changed? And from the first study, when not a single patient got TPA, all the patients in this trial got TPA. So we are looking into whether there might be an interaction with TPA. And the largest study we're doing now is in subarachnoid hemorrhage. And in fact, I won't go through these, but there are now either ongoing or completed quite a number of clinical studies, all led from Manchester, but now multi-centre. And the 2 big ongoing ones at the present time are a phase 2 study in intracerebral hemorrhage, very serious condition, more rare, and a phase 3 definitive study in subarachnoid hemorrhage. We chose subarachnoid hemorrhage because of the most recent stroke study. I could have said also that interleukin-1 has been implicated in many different neurological disorders and indeed in some psychiatric disorders. It's been implicated in schizophrenia, in depression, in many others. In some cases there's good evidence, and in others there aren't. But just as I finish, I do want to say a huge thank you to all the people. I just want to say a couple of words about public engagement. So, large group in Manchester, very large group, too many to mention. I'm extremely fortunate that since I became Vice-Chancellor, they let me meddle, so I can still do some research. Because there are now 8 members of staff at the university who train with me at various different times, 4 of them are professors, and they do all the hard work, and they're both clinical and scientists, and I go to the lab meetings, I read the grants, I read the final papers, and I sort of meddle when I want to. Public engagement, talking about animal research, has always been a focus of what I've done and what this group currently does, largely led by Stuart Allen. They do a huge amount of work with stroke survivors and with the Stroke Association. Many descriptions and discussions. We've had stroke survivors come into the animal facility, see exactly what we're doing. They see the surgery, which can be quite distressing. It's now instilled in all researchers. They all take part in it. Very pleased that Manchester won the only gold watermark from the National Coordinating Centre for Public Engagement, and they talk to schools, the public, and so on. And the animal facility in Manchester is very heavily involved in this and has always been extremely supportive. But I'll leave you with a thought, which, if you remember what I said when I was 2, still aspiring to be a famous scientist, and I was given this present by somebody a little while ago. And it's somebody who knew me quite well, and I thought I didn't quite know what to make of it. It says, well-behaved women rarely make history. I shall finish there. Thank you.
thank you, Nancy, for a wonderful lecture. Seems to me it was a stroke of good luck, not bad luck.
Well,
But anyway, you called it bad luck. I'm not quite so delighted to realise that since I've got a stinking cold, I've got a 3 times higher risk of a stroke next week.
yeah.
And I don't feel quite so happy, but still. Anyway, we've got time for questions, and I'm sure people would like to ask some, so please raise your hand, wave, and the microphones will come to you. All shy, come on, somebody ask a question. Well, while you're thinking, it makes me think, and I should know the answer, but I don't. Since you can give IL-1 receptor antagonist in a significant period after the stroke and reduce the symptomology and perhaps recovery enhances, have people actually systematically tried this post-MI?
Yes.
I know it's a different process, but...
Yes, they have. And it's very interesting. There were studies done in Sheffield, and they went to a phase 2 trial, and the outcome was negative. And then it was found that there was a flaw in the independent analysis, and it actually was positive. It wasn't powered for clinical efficacy, although there was what you always look for, that hint of efficacy. But it certainly met all its primary endpoints after MI. Yeah. And I would think you'd give it in conjunction with the other things you give in MI. Why not? It's not harmful. It doesn't do... so it could be very useful to be given post-MI.
Yes, because I think by comparison with the idea that you chronically give an antagonist to try and reduce the incidence of MI, this is, well, it might work, but it's A, very expensive, and B, doesn't do very much.
It's even been suggested that perhaps it should be given to patients at high risk prior to, carotid endarterectomy or carotid bypass or coronary bypass or transplant, because the last thing you want is an inflammation during that period. So why not just, it will bring down temperature, if anything else.
Yeah. Question at the back.
Thank you for that wonderful lecture. The work that you showed with the multi-centers all repeating the same work is very inspiring, and also that was in different strains of mice and some rats, I think. So that's felt very definitive. Moving forward to patients, did you feel you should also try in other species of animals?
Yeah, we haven't done, but others have. It's never been tested in primates, but it has been tested in pigs, rabbits, and I think at least one other. Yeah, and of course clinical trials are always multi-centre. So the worry with the small studies on patients are always done in a single centre, incredibly well controlled, all under the same conditions. Then you go to a multi-centre trial and it all goes to pot, because it's all slightly different in every one. What I should say, and I didn't go into the detail, is we narrowed down the entry criteria quite significantly for entry into the trial, because that's the other problem. If you take all comers, some of them might have a clot, some of them might be hypertensive, so we did narrow down the range of severity, the range of age, and the time of presentation. But it's not been tested in primates. There was one small study in marmosets, which looked promising, but wasn't very definitive.
Anna, question down here.
Can everybody hear me?
Shout, perhaps. Yes, John.
Nancy, is brain inflammation ever a good idea?
Don't think so. I don't think it is, no. No, let me take that back. If it was highly localised and defined, it could be hugely beneficial in a response to a very localised injury or a very localised infection. That's what inflammation is good for. When it's very, very localised and it's terminated quickly. But if you think one of the major problems in cerebral malaria, in head injury, in hemorrhage, and all of those, it's a really bad thing. But like most things, our immune system is phenomenal and inflammation is phenomenal as long as it's very tightly controlled, and there are many controls on it. It's when it seems to go out of control. But I'm often asked a slightly different question, is why have we evolved to have interleukin-1 produced after a stroke when it's so damaged? You could use the same argument of why have we evolved to have all sorts of nasty things produced. Firstly, you could argue that many neurological disorders probably occur later in life, so there would have been no evolutionary pressure either way. And secondly, there is evidence that very small amounts of interleukin-1 are protective to neurons. So it could be that they're protective. And the final thing, and this is highly speculative, is you could imagine that actually if you've got damaged cells in the brain, which you know well, that are misfiring, that's a really bad thing. You're better off with them dead probably than misfiring. So it could actually be, let's kill these cells. Or it could have been something that emerged in response to cells with DNA damage, as in cancer.
Question at the back.
Hiya, I'm just wondering whether or not there were any differences ever seen between male and female animals or patients.
Yes, in our studies, not absolutely, i.e. in animals it's protective in females. You have all sorts of worries about, if you're using animals, you've got to worry about estrocycline, whether that affects it, and estrogen is protective slightly against stroke, but not fundamentally in terms of whether or not it works, i.e. protects. In patients, stroke tends to be slightly more common in women, subarachnoid hemorrhage tends to be slightly more common in women. Reason isn't known, just not enough studies done to know if the response to interleukin-1 is any different. Of course, women tend to be much more prone to autoimmune disease, so might be generally more inflammatory, much more prone to lupus, to rheumatoid arthritis. Men do get it, but there tends to be a more inflammatory response. And really interestingly, of course, the time when women with autoimmune disease see an alleviation of symptoms is during pregnancy, when they get immune suppression. MS is slightly more common as well in women.
Hello, thank you for such an excellent talk. I actually wanted to ask about your Gold Award for public engagement.
Oh, right.
What kind of impact has holding that award had within the university?
So I think we won it because they came and they interviewed staff and students, picked at random without any staff there, so we couldn't pretend. And the questions they asked was, is this held as important? Is it part of your training? Is it encouraged? And generally they said yes. So I think that was there anyway. I think the Gold Award has done two things. One is the pride, people are very proud of it. But the second thing is when you do find these isolated pockets, and they are isolated, I hope, of the PhD student who says, but my supervisor says I'm too busy doing my research and I haven't got time to go out on Saturday morning and talk about what I do. I said, well, just go and show them this Gold Award, will you? And they say, well, it won't count for ref. I said, yeah, it will count for impact. Believe me, we happen to know Brian Cox's lectures on television got a 4-star in ref. So, well, they doubled the sale of telescopes. So that was a good measure, actually. But yeah, I think it did. And others will win it, I'm sure. But given, I've always been very passionate about it. I say, you're not doing this just because, we say you should do it, or the funders say you should do it. You are funded by the general public. But if you do public engagement, you pretty much get better at doing most sorts of presentations. One of the things in one of the faculties the PhD students are asked to do is talk about their research, and the audience is the cleaners and the secretaries, and they judge. And that has two benefits. Firstly, it's a public audience with no knowledge, and secondly, it makes the cleaners and the secretaries feel very proud and much more engaged in the research that's going on in their building. And we've done similar things with animal research, talk to them about what we do, and then they feel a pride in it.
More questions? Wendy?
When you did the Life Scientific...
Oh, yes. Yes, with Jim.
You mentioned your animal research.
I did.
Quite a lot through it. Did you have any... Did you get feedback on that at all? Did you get any negative...
No, I didn't. 30 years ago I would have done. I would have gone into it. I would have done it, but I would have thought, oh goodness, I've got to think about this. And then, the university would have said, oh, what are you doing? Are you going to talk about it? Oh God, do we need to get you more protection? I didn't really think about it anymore. This is Concord out of Openness. This is what UAR's done, what NC3R's, what everybody else has done. I don't really think about it now. I occasionally get one or two people writing in or Twitter comments, how outrageous this is, what you do. But nothing like it used to be, where there was a sense of real hatred and organised unpleasantness. I don't have special branch visiting me saying, don't park your car there anymore and you need to change your phone number and stuff. It just doesn't really happen. I think I even raised it on Desert Island Discs, actually. But, yeah, I didn't worry about it then. No, I didn't think about it. One back. Oh, Mike. Manchester.
Plant, is it?
Yeah, it's a plant. Yeah. Now he's going to ask me the most difficult question. You said you did this, you didn't really.
How do you explain to the general public the fact that you've done thousands of animal studies over the years and we've still only got one target for stroke?
That's a very fair question. So...
Stuart gets this question.
I know, yeah, Stuart Allen is now leads the neuroscience group. First of all, stroke is very, very complicated and very difficult. You think how hard it's been with cancer to get treatments, how long it's taken, and actually you can get at cancers much more easily. Trying to get at something that happens inside the brain is unbelievably difficult. And I think spent a long time learning, and I think it's best to be honest and say we didn't get it right always. And I think we're doing better now. So to pretend that actually, oh, it was all fantastic and we did everything brilliantly, I don't think is the right way. I think I've always found when I'm asked that question is, yeah, we learnt a lot as we went along and we realised we weren't doing it all as well as we could have done. And then they say, well, do you know you are now? And you say, I can't be certain. The trials might fail and it might still be wrong. And, you know, what do you do then? But I always find honesty's best.
Most pharmaceutical companies now would probably want some indication that there's good human genetic evidence that the IL-1 system is important in stroke. And I guess there is some, but it's not dramatic, is it?
There is some, yeah. I just very briefly skipped over the fact that we've done a lot looking at polymorphisms in the IL-1 family, and others have done as well. And we looked particularly at patients at high risk who did get a stroke. And the two groups we looked at were patients who'd had a TIA, a transient ischemic attack, or a mini stroke, quite a number of which go on to get a major stroke, and ones who'd had subarachnoid hemorrhage and did go on to have a secondary ischemia or didn't, and asked what featured there. So, and we looked for plasma biomarkers as well. We found polymorphisms associated with the promoter region of the IL-1 receptor antagonist gene, suggesting maybe it's not too much IL-1, but it's too little antagonist. And the single correlate from plasma biomarkers was the ESR, erythrocyte sedimentation rate, which of course is an inflammatory marker to some extent. That came out of both studies. So yes, but not great. There are extreme mutations, of course, in the IL-1 family where you get massive inflammatory conditions, and IL-1RA is used in those, in extreme hyperpyrexia and things like that.
Okay. Any more questions? Yes, one here.
You mentioned the liver.
I think we need the mic, otherwise
just
people
didn't
at the back may
expand on
not
it.
hear you.
Indicated there was. Oh, sorry.
It's all right, I can repeat the question.
You mentioned the liver, but didn't expand on it. But also mentioned that there's the impact of the periphery.
Yes.
And that's become more trendy, but also relevant, obviously, in terms of understanding the scalp-brain axis.
Yep. So what you see after a stroke is the classical acute phase response, and many of the acute phase proteins are produced by the liver. And we think, and some of that happens very fast. We think it must be neural, some of it, because you can actually see it in minutes. So it could be neural supply to the liver. And then, of course, the liver activates a load of other things. But bone marrow, an experiment Adam did, he denervated bone marrow in the leg of animals and showed that there was a neural component to that as well. So it's the whole acute phase, stroke is no different to having, breaking your leg. You just get a massive acute phase response in the brain and in the periphery. The surprising thing was that IL-1 seems to be important in the brain and outside the brain. That was somewhat surprising.
I guess my question is a little bit broader than the specifics. Nancy, that was a great presentation, thank you very much. There seems to be, we take a much greater focus nowadays on mental disease as being something that is actually physiologically difficult or pathologically changing perhaps, and associated with inflammation for diseases such as, or conditions such as depression. So is what you're describing here part of that whole story around how we will understand the mechanical changes that take place in mental disease as opposed to just thinking people going crazy?
Well, the first thing to say, in the spirit of honesty, is I'm not a psychiatrist, and so I should be careful here. I think the evidence for inflammation in psychiatric disease like depression is fascinating but not definitive, would be my take on it at the moment. And you have to rule out the fact they've got patients might have something else wrong with them that's causing inflammation and they're depressed about it. So, but there's been some very interesting studies not done by us. I mean, Robert Dancer did an awful lot of work on depression in animals and on behaviour. And interleukin-1 is a very key driver of what's often called sickness behaviour, which is the fact that you'll feel depressed when you've got some sort of infection, you're sleepy, you don't want to eat, and that is all IL-1 mediated, or at least block it, and you do block it. What hasn't been done very much, and of course minocycline, anti-inflammatory, is in trials in quite a number of diseases, mental health diseases. I've not seen any really big studies, and that's not to say there haven't been any, because I don't follow it that well, the literature, to show that actually there is a very strong correlation between depression and bouts of depression and inflammation or the anti-inflammatory agents. And it's very hard doing these studies because the first research on dementia arose because of the lower incidence of dementia in patients with rheumatoid arthritis, but only if they were on anti-inflammatory agents. Then they did the proactive trials and they couldn't prove it. Now they've done them better and for longer, and it is looking like it's true. So they're quite difficult studies to do, I think.
Okay. If there are no further questions, then the first thing I have to do is make sure we all thank Nancy again for a splendid and don't worry, I'm not giving more speeches. I want to say something which isn't in my notes. I want to make sure that we recognise the whole of UAR staff who've done a fantastic job, A, in running and keeping UAR going as the wonderful organisation it is over the last year, but also in what they've contributed to putting tonight together. So thank you everybody very much. And last of all, if you exit the lecture theatre and turn left where you had tea and coffee, there might even be a drink or two for you. Thank you.